Indian Botanical Society. Year before, year before last, she gave J.C. Sengupta Endowment Lecture organized by West Bengal Academy of Science and Technology. She has published more than 200 papers, scientific papers, as communicating author and has 3 patents on her name. She and her able team made remarkable contributions in wheat biotechnology and ceri biotechnology. As part of the international initiative, she has contributed in sequencing chromosome 11 of rice and chromosome 5 of tomato. Her group has sequenced the complete chloroplast genome of mulberry and presently sequencing chromosome 2A of wheat. I'll conclude here only without buying much of your time, as if all of you are eagerly waiting to hear Professor Paramjit Khurana. Now, I'll request Professor Khurana, Paramjit Khurana, to, to deliver her lecture.
Thank you very much, Dr. Pal. Indeed, it's a great honor and a privilege to be conferred with this particular award because I happen to know both of them. I had the good fortune of interacting and knowing both of them, Dr. Archana Sharma as well as Dr. A.K. Sharma as well. In fact, both of them were frequent visitors to a lab when I was doing my PhD with Professor Maheshwari at the Botany department. And I remember in my initial days when they used to come to the lab, we used to discuss our problems with them. And after a couple of years, again, when they came back, Dr. Archana Sharma, she remembered me by my name and she asked me about what progress I was now making with respect to the project which I had discussed with her last time. So that I was so impressed with her memory that a person working in someone else's lab, she would remember that not only in the name, but also the project and the problem and what were my concerns and how I was coping up with them. So I was really very, very happy when even the Science Congress awarded me the Archana Sharma Award quite some time back. And then I've had the good privilege of even having the Nasi Archana Sharma Award here. And I feel that as if I know her so well personally. And that is the reason I was looking forward to coming and personally delivering this lecture. But unfortunately, because of the circumstances which developed last couple of weeks, I had to cancel my program, which was already scheduled and made up. But anyway, I would take this opportunity to thank Dr. Amita Pal, of course, the president, the Anita Mukherjee also, the vice president, and Dr. Rita Kundu also, whom I've been troubling quite a bit here and there, and the head of the department of the botany department, Shantanu Paul, whom I was wanting to meet again in person as well. So in fact, it's a great privilege and honor for me to deliver this talk. And if I can start sharing my presentation, post disabled participant screen. Okay. So if I can share, no, I need to be able to share my presentation. I think if you can give me the, okay. Yeah, I can do it now. Just a minute. Yeah, is the slide clear? Is the slide visible? Is the presentation visible?
Yes.
Okay, good. So the title which I gave for my presentation was Plant Genomics and Climate Resilient Crops. Because whatever we are doing, I think we need to be able to translate that into practical utility and see how things work up here and there as well. So this lecture, I would say, is in the memory of Dr. Archana Sharma and Dr. Arun Sharma, both of them, who work not only with cells as cell biologists, but even they work with the nucleus and what is internal in the nucleus. At that time, they were considered the best in cytogeneticists. But now later, then they even tried to get into the details of the variation which is present in the genomic DNA content with it. So from here, I think it is where we would be able to see how we can go forward from what they laid the basis of it and how we have taken this further. And I would like to say that it was great interacting with both of them. They were stalwarts in their own field. And I wish I had the opportunity of delivering this lecture in their presence as well, because there are very few people who are interested in science as much as this couple was, I would say. So today, moving forward from the DNA, I think once when earlier we used to talk of DNA, that is a fragment or a strand of it. But now has come up into practice is genomics. That is the entire DNA complement of that cell. Similarly, when we used to earlier talk of the RNA molecule, we are now talking of transcriptomics. That is, what are the different types of RNAs which are getting upregulated at that particular point of time in that particular tissue. And similarly for proteins as well, I would say the entire, not just studying one protein, but the entire protein complement of that cell and that condition. And that discipline is now termed as proteomics. So basically, we are in the era of omics, I would say, whether we are working with DNA as genomics or transcriptomics or proteomics and metabolomics. And all of them, they give you different sort of information which is happening. In fact, when you're working with genomics, it tells you the potential of what all can happen in that cell. While when you're dealing with transcriptomics, it tells you at that particular time point as to what is happening. While for the proteomics, it tells you what has made it happen and what makes it happen also. And similarly for metabolomics also, what has happened and what is happening at that particular time point also. So depending upon what is your insight into it, you can get different types of information related to these disciplines. Now, this we say is a very good time for plant genomics because fortunately for us, country India participated in the International Rice Genome Sequencing Program. And India became part of it. And this work, yes, was published way back in 2003 and so forth. With India's expertise in rice, we were invited to be part of the International Tomato Genome Sequencing also for various reasons. And we have also been part of the Wheat Genome Program too, which has also been completed by now. And in-house I say, because this is not international collaboration, but a lab alone has been associated with Mulberry. I need not introduce Mulberry to Calcutta because here people know what Mulberry plant is. But we have worked on the chloroplast genome and the nuclear genome of mulberry as well. Simultaneously, we have also collaborated with our colleagues in the microbiology department and zoology to work on the genomes of the special microbes present in the indigenous conditions of our country, especially in the hot springs of Manikaran and so on. So this is a snapshot to show you the map-based sequence of the rice genome, which is considered a landmark. This work was published in August 2005, while the deadline was December, but it was published a couple of months earlier to that. Why this work is considered a landmark in plant sciences is because not that it is just a sequence here, but this is considered, the sequence quality is still today known as the gold standard quality, which means that there is less than one base pair error in 10,000 base pairs also. That means to make it that good a quality, you have to sequence that same strand of DNA redundantly n number of times so that the error rate is gone. So besides the model plant Arabidopsis, there isn't any other gold standard genome known so far. And rice being a model crop plant, it is very fortunate that this has been done. And why I consider this as a landmark, not only internationally for plant sciences, but also for the Indian sciences is that India for the first time participated in an international effort. Although at that time, we were partly responsible for sequencing of chromosome 11 alone, because you can see here, different countries were given the different chromosomes to sequence. Now, what happens is, in an international program, you have to work at the same speed. You have to follow the same standards, same cutoff values, and so forth, so that finally you can collate the data together. And interestingly, all the genomes of all the 12 chromosomes of rice were collated at Delhi University in a very small room. And this was a very great learning experience for us all also, because we had never ever done all this so far. So this was a great experience, not only otherwise, but otherwise too. So this work, which was published in Nature, also got covered in Science. And I bring in this picture to show you, this is the annual meetings where the PIs of different countries are being represented. And I bring in specially to point out this particular person, Sasaki Furuya, Sasaki, sorry, who spearheaded this particular program. And he insisted that this data be available in the public domain, not in the private sector domain. That means irrespective of whether your country has participated or not, you will be able to use this work. And that is what actually happened after the sequence of the rice genome. The functional genomics and the gene identification in rice saw a log curve. That so many genes of interest of agricultural importance, say like seed shattering, seed number, starch biosynthesis, seed dormancy, gibberellin insensitivity, shoot meristem identity, silicon absorption, fungal bacterial blight or fungal resistance, rice blast, many of them, early heading stage, heading date, and so forth. All these genes were functionally, they were identified and they were functionally analyzed also in rice in this particular case. That is the beauty of the availability of this sequence and how this could be translated into this. At that point, for our own sake, in the next year, we sequenced the complete chloroplast genome of mulberry, which happened to be the first complete genome to be sequenced from a country. And this was a very good, satisfying experience for us to say that, yes, we could do this particular exercise as well. It took us about a decade to get funding for the nuclear genome of mulberry, and I wanted to do that as well. And we've been able to finally do that, report that in 2022. And this shows a comparative analysis, and we identified about 21 accessions, resequenced them. We identified 2.5 million SNPs and millions of indels. And also we combined the genome with the transcriptome as well, and identified species and tissue-specific genes also in mulberry. So this was also a very good ending point for me, I would say. But the rice genome, slowly, because of India's expertise in the rice genome, we were invited to be part of the tomato genome consortium also. And this is because tomato, it was thought that this is a fleshy fruit and you would be able to analyze many other traits besides the traits which were present in rice. And this work also got published in 2012 or so. Now, interestingly, if you see, there are more than 7,000 varieties of tomato which are present here, but there is very little genetic variation at the genetic level, okay, at the genomic level, which means that most of the variation is occurring at the transcriptome level and so forth. So how can we extrapolate this information? And in fact, the tomato genome program soon transformed into the Solanaceae genome project, okay, the entire family of Solanaceae, because if you see here, in members of the Solanaceae, you have not only tomato, you have chilies, you have potato, you have sweet potato, you have horticulturally important plants, you have medicinally important plants, you have most of your vegetable crops, they belong to Solanaceae. But most importantly, I think, you have genes for adaptation as well, because members of Solanaceae can be found in the cold desert and the dry desert area also. That means it was thought that if you had the tomato genome sequence available, you could be able to extrapolate this by comparative genomics to other crops which are present, which had different types of interest and characteristics which are present here. And that is why the name became as the Solanaceae Genome Project. And in fact, by virtue of comparative genomics, much of this information from tomato was extended to these medicinally, the horticulturally important plants, to the starchy crop plants, and to the other crop plants also. At the time when the Rice Genome Program was listed, the project which was initially listed was for wheat, but at that time it was believed that the wheat genome would never ever be sequenced. Why? Because number one, it is a huge genome as compared to the rice. The rice is about 420 megabases, but the wheat genome is about 16,000 MB or so. Plus, it's a hexaploid. It does not have two parents, it has three parents. And at that time, technologically, we were not capable of handling a complex genome like the wheat genome. But subsequently, what has happened is with technological advancements in the sequencing technologies, which I'm not getting into the details, this work also was realized. Now you have next generation, second generation, third generation, and other sequencing technologies which have come into existence. But in 2018, I think we published the rice genome, the wheat genome as well. And in here, again, there is a comparative map of all the transcriptomes of different tissues and the different genomes which are present in this particular condition. Okay. So now moving on to how this genomic information can be put to use. What are our challenges, agriculturally important ones here, and how can this information which is generated can be done. So if you see simultaneously in the field of agriculture, we have moved from green revolution to the gene revolution, and we are now in an evergreen revolution kind of a situation. Okay, so looking at the food security challenge which is available and what the world would be feeling. So in 2050 or so, I think there are 3 A's which become very important for food security challenge. That is, number one is availability. Availability of proper foods in different geographic areas. Its accessibility and its affordability. I think all 3 are equally important to make a nation a food secure nation as well. And if you look at agriculture, especially in India, because our economy is totally agriculture dependent, agriculture is the backbone of our economy. But this agricultural practices also affects the environment in different way. We generally don't think of it that way, but for agriculture to succeed, number one, you need deforestation because you need plains which are present here. You need land for monoculture to come into practice. Plus, when you're spraying the pesticides and the nutrients and the fertilizers and others, these chemicals also impact the environment in an adverse way also, besides their intended users. And that is why the newer terminology which is coming into existence is climate smart agriculture. That is, irrespective of what your land resources are, your water resources, and your bioresources which are present here, you should be able to manage your ecosystems for a sustainable livelihood. Now, this is a small sentence, but it has major challenges to it. And some of the biggest challenges are that we are actually outgrowing the earth. What do we mean? We mean that the food security challenge is leading to a decline in the water tables and a rise in the temperatures over a period of time. And these are the 2 major environmental factors which you have to deal with when you're dealing with agricultural productivity. One is water tables and the other is the aerial temperatures which are present here. And we have tried to address both of these through our studies, try to understand what is their behavior and how we can handle them differently, especially in our croplands and so on. Now, coming to the first one, that is water. Water, I say, is a lifeblood. I say it's a lifeblood because if you see, this is one substance, one resource, which has no substitute. You cannot replace water with anything else. So it is very essential to be able to utilize our water resources in a judicious manner. And if you look at the water which is available on the Earth planet, we have about 97% is water, but it's only 3% which is fresh water. And it is this fresh water which is fit for agricultural utility and not the other saline water. And out of this 3% also, most of it, majority is present in the ice caps in the form of glaciers and ice caps and so forth, and very little is present as groundwater. So basically, you have 1% of the fresh surface water which is available for agriculture to be used, and that is why it is important that we use this efficiently. This is, I show you a satellite picture of before we started our work on water sustainable water utilization by different plants, by different approaches, especially by biotech approaches. This is a satellite image to show you how the groundwater is getting depleted at what rate. If you look at the scale here, the red indicates more than a meter per year. Okay, and this is about in 2006, 5 or 6 or so, the satellite picture which is present. And if I enlarge this area, this particular area, which is boxed up here, which is India is present here, you'll see here that it is here in Rajasthan, Haryana, and Punjab. This is the region where the water table is receding at more than a meter per year. And this is known as a food basket of a country because you have maximum crop productivity of wheat and rice at this particular region as well. Now, I'm sure you've all heard of the carbon footprint. A similar terminology exists, which is known as the water footprint. That means how much liters of water does that crop consume to produce a kilo of that particular grain material, if we talk of only food grains. Like for corn, it needs about 450 liters or so. For wheat, 500 liters of water to produce 1 kg of wheat. Look at rice, 1700 liters. And if you look at the recent pictures, the newer varieties, it's about 2300 liters or so of water is consumed to produce 1 kg of rice. And that is why in our earlier days, rice and other plants which had a higher water footprint, they were cultivated in the coastal regions and not in Punjab and Haryana, where water was limited. There it was wheat. But the moment you start shifting the agricultural practices here and there, it will have an adverse effect on the water tables also. And I say this water molecule, although it's a very small molecule, chemically it is H2O, okay? Just H and 2O, which are present here. But it is so important, and the plant, which is totally dependent on it, regulates its use beautifully. The plants are very good regulators, depending upon the soil supply, the depth of the roots in the soil temperature, what is the soil moisture, what is the soil texture, and the atmospheric demands. How much is the sunlight? What is the air temperature? What is the humidity? What is the wind speed? The plant regulates it very nicely. It's water use efficiency, and it is known as the water use efficiency. Now, what happens is we were working on a different plant, and we had this one particular gene, which I'm not telling you all the details because it's a lot of work, but you can take my word for granted. It is a Hordeum vulgare. It's a agglutinin from Hordeum vulgare, and we were working on it for introducing drought and salinity tolerance in it. Okay, we were working in wheat, but at that time we were approached by our colleagues at the Sericulture Institute in Mysore, which has the headquarters for mulberry. Okay, it is in the south that you have mulberry plantation growing on. Now, this is a field of mulberry, and mulberry is the second fastest growing plant after bamboo. It grows very fast. It can grow up to a meter per day. Okay, a feet per day, not a meter, a feet per day. And it uses a lot of water. And that is why in the south, where there is abundant water, it is grown. Now, once it is grown, it's a tree maintained as a herb. Okay, so it is pruned regularly, and once you plant the crop, it is there and stays in the field for 5 to 10 years or so. So it needs a lot of water. So when the sericulturists, they came to know that we were working with HVA, they said, can you help us also make mulberry which will grow under less water? So they can grow it under water deficit conditions as well. And to again make a big story short, we introduced in this HVA gene by transgenic approaches in mulberry, and we raised the transgenics, characterized them, did all the background work here. This is the non-transgenic plant. If you do not water it for a week or so, it starts wilting, but the transgenics will not wilt. Now, another important thing with mulberry is it is the leaf which the silkworm feeds on. And the leaf is plucked and given to the silkworm. And if you ask the sericulturist, what are the qualities in this leaf which the silkworm prefers? They will say the leaf should be nice and succulent and nutrient rich. It's a very nice interaction, gene for gene hypothesis, which has co-evolved between the silkworm and the plant also. The silkworm will not eat the leaves of any other plant, and the mulberry is grown only for feeding the silkworm. So what happens is when you pluck the leaf and give it to the silkworm, the water content drops. So we did not know how to translate this nice and succulent leaf quality into analytical. So when we checked for the relative water content in the leaves which had been plucked, decapitated from the main plant, and if you look at the blue bars here, if you leave it for 20 days, water content reduces in the leaves to about 40% or so. Whereas in the transgenic plants, it is still at 75% or so. So we could make a plant which will tolerate better even in the absence of water content which is present here. We did not water it for 20 days or so, but at the Sericulture Institute, when we transferred our transgenics to them, they did not water it for more than 2 months, wherein if you do not water, all the mulberry dries except for a transgenic mulberry, which did not dry. And these transgenic mulberries were tolerant to both not only drought, but also salinity as well. When we did the morphometric analysis, this is just to show you that under constitutive promoter, yes, there was a little compromise on the leaf quantity, but when we used a inducible promoter, then the leaf quantity also doubled up under stress conditions, water stress. We introduced in many other genes also for them, especially for biotic stress tolerance against fungi, because it uses lots of water, so it is prone to fungal infections and bacterial infections for leaf blight, for anthracnose, and for black spot diseases. And we show that these are the controls and these are the transgenics and how well the transgenics do under different conditions. In fact, in Ladakh also, they grow mulberry, but for different purposes. And we had introduced a beta-carotene hydroxylase gene for them, which would give oxidative stress, protection against oxidative stress of high irradiance, high temperatures, and even UV stress tolerance, because at the higher altitudes, you find more of UV radiation, which is present here. So we had different transgenics with different traits available, transferred them, we reared them, characterized them. We did all the analysis in the transgenic in the greenhouse facilities at South Campus, and then we transferred them both to CSRTI Central Institute at Mysore and to the University of Agricultural Sciences in Bangalore, where they are thriving, and they did the field test, and they also did the biotic assay with different other pathogens of mulberry, and what the silkworm, how much is the silk quality, and so forth. And we find that there is no compromise on this. However, this work is here only in the experimental state. It never went for public utility, although this plant can be vegetatively propagated. It could have been transmitted anywhere because of biosafety guidelines at that time. We could not release it to the public as well. So we shifted our attention to the other important crop plant, that is wheat. And wheat, as I mentioned a while ago, is a hexaploid. It has 16,000 MB as the genome size. It has 3 parental genomes known as the A, B, and D genome. And at that time, for a molecular biologist, no one would work on wheat. Why? Because more than 80% of the DNA was repetitive DNA. There were hardly any ESTs. There were only 100 ESTs known per megabases of the genome. While in rice and maize, there were about 3,000 to 500 or so. And yes, it is susceptible to abiotic stresses also, besides the biotic stress factors. And people thought that it's a nightmare for molecular biologists. And 2-3 students of mine also were very frustrated when they started working on wheat for various other aspects also. However, we were very lucky in having some very good students, and we had transgenic wheat with us, not only of the hexaploid wheat, the bread wheat, but we also had for durum wheat, which is a tetraploid, and emmer wheat, which is a diploid wheat, this is also a tetraploid. Emmer wheat, the diploid one is dicotyl. We had another one also. We had both for diploid, tetraploid, and hexaploid wheats which were present, but we could never take them for analysis. Now, in fact, our first work was on cereal cyst nematode way back in 2003, wherein we had shown for how, by introducing a gene of interest, we could introduce in cereal cyst nematode tolerance. Despite the fact that I was invited to CIMET and Mexico for presentation of this work, because till today, I'm saying we do not have a source of resistance for Heterodera aveni, which is the cereal cyst nematode. This work being transgenic in nature was not popularized, but the proof of concept was given by us that yes, you could engineer for cereal cyst nematode as well. This work was done in collaboration with our colleagues at IRI, who is an entomologist, and she did all this work for us. So we were stuck with transgenic wheats, we were stuck with biotic stress wheat, we were stuck with abiotic stress wheat, and we could not take them to the field because we could never do the homozygosity tests and we could never do the progeny analysis because we would get fewer transgenics and we need more population to do that. So we hit upon, one of my students hit upon another thing. He says, why don't we make haploids in wheat, transform them, diplodize them, make doubled haploids so that the entire genetic complement gets stabilized. And we did exactly that. This is to show you how we got the microcal, although the success rate is only 2%. But yet we could recover the transgenics, which were doubled haploid. We characterized them for abiotic stress tolerance. And you can see here, even under simulated drought conditions, while the transgenics will not grow, even at 400 millimolar of mannitol, you can see that mannitol or PEG, I don't remember one of them, but the transgenics will grow. But then, as they say, the proof of the pudding lies in the taste. And in wheat, it is the productivity which is to be seen here. So we also studied many of the maturity-linked traits and crop yield in transgenic wheat as well. And we showed that if you look at the single seed weight and the seed weight per spike, the percentage reduction is about 50% or so in the non-transgenics, whereas in the transgenics, the percentage reduction is about 20 to 25% or so in this particular case. And this work was also mentioned very well. So we had transgenic wheat, which was drought and salinity tolerant. And herein we had doubled haploids in wheat, and there was no problem. And we had multiplied them. And we were hoping that maybe now we would be able to distribute it to the public, but then there were no takers for it because of its transgenic nature as well. And this remained a wishful dream for us. But then what did we do? We had this doubled haploid wheat with us, which was now at the 8th and 9th generation, breeding true to type. The entire genetic complement is uniform, except for the gene of interest, which has been incorporated. So we tried to check and see what effects a single gene would have in a totally homogeneous background, genetic background or so. And recently we've tested these also for heat stress tolerance. And this work has also been published in Cell in 2022 also. And you can see that the entire cell undergoes a homeostasis. Okay, that is about the transgenic wheat as well. So when we realized that we were getting nowhere with the transgenics not being able to release it to the public, we shifted to the basic research. We said, let's do what we can do and contribute to the information generation, as I say, creating more information. And then looked up at wheat because in temperature is very important. In case of temperatures, it is seen that in the wheat growing bread basket of a country, the temperature in 2006 rose more than 5 degrees or so. Okay, in 2006, and we had not yet started this. This was the earlier data. Now, why this 5 degrees made a cause of concern? It was a big cause of concern because this increase in temperature occurred during the months of February to March. And in fact, it was not 5 degrees, it was 7 degrees above normal temperature. And this period in February and March is the most crucial for wheat because this is the time that the spike emerges from the vegetative tissue. Okay. And every degree which increases in temperature is said to bring down productivity by 4%. For all crop plants, but for temperate crops like wheat, this productivity downfall is about 6% or so. So you can do the calculation. In 2006, the temperature was 7 degrees above normal, and every degree brings down productivity by 6%. How much wheat did we lose just because of heat stress? More than 40% or so, only because of heat stress. We're not talking of the other things. And unfortunately, in 2007, India, that was the last time India had to import wheat. So it was a good warning for us to understand what happens in wheat, how heat affects wheat, and so on. And then we realized that while in the northern states, in Haryana, Punjab, and others, we talk of the heading stage, which gets affected by heat stress. In the southern stage, the wheat plant is still at the tillering stage. That means it is making more than one vegetative shoot here. So whatever will be the mechanism of adaptation or resistance or tolerance, rather, I would say, for heat stress, which works for the northern wheat varieties, will not work for the southern. We need to have a different mechanism of tolerance introduced into these varieties also. Besides this, we also realized that fertility is severely compromised under heat stress conditions. So we realized we had to target the reproductive tissues as well in wheat, both the carpel and the stigma as well. And at that time, in 2006, the International Cretaceous Mapping Initiative said that the first and the foremost objective is to produce ESTs, that is the express portion of the wheat genome, so that it can, you can have screening, normalization, sequencing, data analysis. You can get into deletion mapping, comparative mapping, and many other things. So this was the main mandate. And we also aligned with the international mandate. And again, to make a long story short, we addressed all those stages, the vegetative stages, the heading stages, and the reproductive stages also. We made the cDNA libraries, sequenced them, made the subtractive libraries. And what did we see after? We saw that there were lots of transcription factors which were getting switched on. There were lots of post-transcriptional control mechanisms in play. There were translational checkpoints in play. There were post-translational control points, and there were many others. In fact, the entire cell undergoes a homeostasis, gets reorganized with respect to heat stressors. So this made our task even difficult. And in one of the international conferences in Russia, when I presented this work at St. Petersburg, this famous wheat breeder comes up to me and he says, young lady, now you have a big challenge ahead of you. You need to tell me what is the function of each and every gene, which you have showed is differentially expressed in these tissues. And we took up that challenge in a very sincere way. And over the last decade and a half, I would say now, we've analyzed and functionally characterized lots of heat shock proteins, which are differentially expressed, characterized each one of them and shown how they, when you overexpress them, they can provide heat stress tolerance. We have worked on the heat stress proteins. We have worked on various transcription factors, HSF2D, which are the super transcriptionally regulators, the zinc fingers, the MADS-box, the BZEPs, various intracellular transducers, signal transducers, B2 protein, VAP protein, LTPs, LTP12, STI, even BRI, and various osmolytes like MEPS, MEPS1, MEPS2, and very other miscellaneous ones which are associated with methylation and so on. So this gave us a lot of publication. There was a lot of work, very satisfying to see what is the end product of it. But the end of the day, when I presented this work at one of the international wheat annual conferences, this young person comes up to me and tells me, ma'am, can you help me in the Indian context? What do I know? I don't know which are the varieties which are heat tolerant and which are heat sensitive. And what will I do if my crop faces a heat stress in the field? So we started looking at these basic questions also as to how our data could help these people also. So first and foremost, what we did is we made a heat susceptibility index. We characterized some of the 12, 13, maybe 18, 19 ruling varieties of wheat and looked at their seedling stages, their FV by FM values, the membrane stability indexes, the temperature induction responses, the carbon profiling, the proline and others. And we said that these are the varieties which are relatively tolerant to heat stress, while these are the ones which are relatively more sensitive to heat stress. That's all we could say at this point. That means we tried to characterize the ruling varieties and say which were more capable of tolerating heat stress and others which were not able to. In a parallel project on BRI, which we were studying, we also found some very interesting things when we extrapolated that to wheat. Now, these are the grains of the two tolerant varieties of wheat and two sensitive varieties of wheat under normal conditions. This is how the grain looks. I don't want to give you the analytical details and showing you the pictures. And this is what the grains look when they face 5 days of heat stress in the field. Okay, this grain gets transformed like this here, and this here. I'm sure we will never ever buy this grain from the market while we went searching, shopping for this particular. And this is how the wheat grain gets affected by heat stress. And now when we had this chemical with us from another study, when we sprayed that to the field, which has exposed to heat stress, this grain got transformed into this got transformed to this. They may not necessarily be exactly identical to the original, but they were much, much better than what they were earlier. And in some of them, they were even better also, both for the tolerant varieties and for the susceptible ones. Unfortunately, this is a very expensive chemical and we could not give it for field use. We could use it in the lab to show that, yes, this is possible. And we were looking at engineering for the production of this gene, this hormone in the plant so that you can do that. We also looked at other heat adaptive traits in crop plants, and we focused on the radiation use efficiency because we are looking at carbon fixation. Everyone talks of Rubisco specificity and regulation. We were working on spike photosynthesis, membrane stability, and other things under heat stress conditions and how the light is partitioned. We also took help from the wild relatives of wheat. I said a while ago, wheat is a hexaploid. It is a hybrid of the AA, the tetraploid, with toshai, which is a diploid one. And another one is the diploid is the monococcum, which is present here. I'll show you in a while. One of my visits in June and July to Punjab Agricultural University, where it was very hot, the temperatures are 45 to 47 degrees in the field. I could see Aegilops toshai and Aegilops peltoides growing fine and looking nice and green and flowering also. What did this mean? This meant that these wild progenitors of wheat had heat tolerance in it. Because when I gave them heat stress for 5 days and Delhi University South Campus, one of them survived, even flowered, the spikes coming out, but Speltoides did not survive that well. So we looked at many of the energy distribution, radiation use efficiency of these plants, of toshai and Speltoides. And if you look at it, the top panel is under control conditions and this is under heat stress conditions. In case of toshai, the PS2 activity, that is photochemistry, it gets reduced from 76 to 73%. Rarely, that means negligible. But what increases is the NPQ. NPQ is the heat energy which is dissipated, okay, and the fluorescence also which is present. But look at its Speltoides. Speltoides, which was more sensitive to heat stress, the PS2 activity reduced from 76 to 23%. And the NPQ, which was less than 1%, climbed up to 35%. That means it was radiating, the radiation had more major impact on the photosystem 2 of Aegilops peltoides as compared to in toshai. So we said, so our target was toshai and we should look for heat tolerant traits here. We looked at the same phenomenon in the cultivars also, which we had categorized as relatively as heat tolerant and heat sensitive. C306, which is a drought tolerant variety, was also, we said, finding it as a heat tolerant one. And even here, if we look at the PS2 activity is relatively still in the 70s, while the sensitive one, the PS2 activity reduces to 1/3 or so, like Aegilops. So the same principle was getting extrapolated here as well. And the NPQ was increasing, and so was the other one factor which is present. So what we did is we also looked at the differentially expressed genes in the spike tissue of these under different conditions. And what caught our attention, one of them which I'll mention is the Rubisco activase B and A isoforms, which are getting upregulated in heat stress and downregulated under heat stress in different conditions. So two isoforms of the same gene, one getting upregulated, one downregulated. When we validated many of the other members of the photosystem 2 complex in wheat, I would say, this is for RCA, which is getting consistently downregulated under heat stress conditions. This is our heat tolerant ones, and this is the sensitive variety. Look at the other component, the other isoform. RCAB is still downregulated in the sensitive one, while in the tolerant one, it is getting upregulated. So we knew from where to fish the isoform if we need to introduce this gene into other crops for heat tolerance. And that is precisely what we did. We also looked at the expression profile of the RCA gene family in the leaf, the spike, and the grain, because we need resistance, we need tolerance to heat stress at these three major conditions here. And I said, this is a hexaploid. So we have so many homologs, so many isoforms which are present here in the leaf, spike, and here, but it is these two of them. This one, which is active in the leaf, in the spike, and the grain, is the one which we took for expression in rice. Okay. Rice, when we introduced the wheat gene in rice and we saw the relative expression under control conditions and heat stress conditions, you can see in the transgenics, all of them going up. And when we had you looked at also the Fe by Fm, which we consider as a hallmark for photosynthetic efficiency for Fe by Fm. And if you see here in the non-transgenic, it goes down, but in the transgenics, it is still relatively high in all of this. When we look at the reproductive status of these, there were some transgenics, especially these two, when we calculated the total number of florets and the sterile florets, which are present here. Sterility was reduced and fertility increased more than the wild type also in some of the transgenic. There was one transgenic which we could not understand why this behaved, but yes, you do get sometimes different kinds of things. So this was very, very satisfying for us to show you that this RCA B homolog, when we introduced that into rice, it could make rice tolerant to heat stress conditions as well. Very difficult to continue with the same work an extended period, but we also ventured into the epigenetic regulation of heat stress. What happens if the plant faces heat stress in the earlier stages? Will it remember that information at maturity and so forth? So there are different phenotypic variations for them, for epigenetic variations, and you can study different epigenetic markers for that, and you can model their things here. And we did a little bit of this work as well, and we tried to understand the methylation gene family as to how these the methylated genes have evolved over the different periods of time. But this is more for this kind of a thing. So basically what we were looking at is, we were looking at a regulatory network which is involved in heat stress response. And we had worked upon many of them, the master regulators of transcription, the transcription factors, many of them, which activated them, stabilized them. And so basically most of these traits were associated with acquisition of thermotolerance, how thermotolerance is acquired. We did venture a little bit into histone modifications and microRNAs to study what is the heat stress memory also, how chromatin can be remodeled into it, but not too much, because this is something which needs to be taken up further for long-term adaptation to heat stress also. This is where we've been able to contribute, fill in the gaps here, and show how each and every one of them is interconnected and regulated under heat stress conditions. Another thing which caught our attention, looking at the non-transgenic approaches to introduce heat tolerance, was in the area of rhizosphere engineering. This is when we came across lots of papers with we talk of the PGPRs and the beneficial root microbe interactions. We realized that the PGPRs can aid on multiple ways, They're plant growth promoting bacteria. That means they produce phytohormone, they help in nutrient uptake, they help in biocontrol. We wanted to see whether they will help us in abiotic stress tolerance as well. And we did some initial experiments too, and with respect to heat stress tolerance, got hold of many of the bacteria, but that work is not yet published. But we have some very good, interesting leads here to combine the transcriptome in presence of PGPRs and without the presence of PGPRs under heat stress conditions and so forth. And we are trying to fill in the blanks in this particular area as well. And hopefully we should be able to publish this year or maybe next year or so. It needs a little more refinement in the data to be done. So basically what we've been what I've tried to impress upon you is how we have tried to bring the outcomes of genomics, translate that for higher genetic gains. It is starting from genomic approaches, how that information can be translated, whether it is as a genetic resource itself, different genomic resources have been created as a reference genome or whole genome resequencing, or most importantly, functional omics, and how that can be translated to bridge the gap between a genotype and a phenotype. So you could do genomic selection, you could use it as diagnostic markers, you could identify superior traits here, you could do the genetic analysis. Basically, it gives you information to make informed decisions as well. So these are decision support technologies, depending upon where you are and what area you want to cross from here to here, and so forth. So coming to the end, I would say, I don't want to load up too much on the work here. I would say it has not been a straightforward pathway, certainly not. We thought we would reach from this end to this end, but life gave us lots of twists and turns. And I think what is important is that as long as we know that we are headed in the right direction, that is good enough. It is not necessary that we make a map straight away. If we have a problem, we need to realign our goals, redefine our goals in a proper way, and build. It's important to build a compass than a map, I would say. And that is precisely what we've been able to do a little bit in a small way at some camps. So thank you very much for your attention. And thanks a lot to all the people who have supported us this work directly, indirectly, and all the workers, my students and others, because without their effort, this work would not have been possible. So thank you very much for your attention. And I would be very happy to clarify some doubts if there are any, and if time permits, I would love to do that as well. Thank you so much. Over to the organizers, I would say. Hello. Host is muted. Please, can the host unmute and speak? Hello.
I'm now handing over to Professor Amita Pal,
Okay.
President, for felicitation of the speaker. I think she's very camera.
Excellent presentation, Param.
Thank you.
Very nice, very nice.
Thank
Your last slide shows the path you have passed through so many decades, starting from genomics to transgenics to epigenomics, finally to rhizosphere engineering, the most upcoming area of the research.
Yeah.
Wonderful. I wonder how did you cover all these things?
It is what was feasible, what was doable. It was very difficult, very challenging, but they say when life gives you curveballs, you got to use them in a different way altogether. That's all I can say.
I'm curious to ask a boring question.
Yeah, please.
What will be the fate of the transgenics? Your transgenics, so much
Nothing.
labor you have.
No, I don't see any light at the end of this tunnel here, and that is why I don't even think about it now. See, if I had done that, I would have ended up like another Dr. Deepak Pental,
Exactly,
he spent his life, put his life effort into it, and till today, he's not been able to realize it. So I thought, why, instead of following that path, let me do what is acceptable to the public. If they don't want to invest in biotech crops, we have shown that, yes, this is possible. At that point of time, it was very high because India had just released BT cotton. So we thought mulberry would be another piece of cake, It is not for human consumption. It goes for the silkworm. And you can use it as a green manure and other things also. Grow it onto the wastelands and so forth. But it didn't work out. And wheat, yes. In fact, after we had a wheat transgenics, we were told by the breeders, please don't mention that you have transgenic wheat because it can affect our export also. So there are many times, there are other non-scientific concerns which hinder your progress. So, and I realized very early in time that it was not possible for me to fight that. I could end up fighting against it my whole life without coming anything. So I thought, might as well use my resources elsewhere and do something which may be of interest to someone else someday. So doing basic science sometimes
Thank
is
you
much
very much
more
for this
meaningful.
wonderful talk. We enjoyed a lot. I
Thank
would
you.
be very happy if you could come and present here.
I wish
But
I was there.
virtually, I am presenting this memento and will be sending this to your place later on.
Okay.
Okay? And thank
Thank
you
you
very
so
much.
much. Thank you. Thank you so much. I'm really indebted to all of you and sorry for having troubled you all with all the exercise, but I was hoping that I would be there in person and get more time to interact. It's not just delivering the talk, it is the interaction with the senior faculty and the youngsters also, which I was looking forward to. But anyway, maybe some other time.
Yes.
Yeah, we
Thank
are
you so
also
much.
looking forward to that opportunity, ma'am. Can I now request Professor Rika Kundu to deliver the vote of thanks?
My heartfelt reverence to Didi and sir. Good afternoon, everyone. On behalf of Archana Sharma Foundation of Calcutta and on my own behalf, I extend my heartfelt gratitude to each and every one of you for gracing the 8th Professor Archana Sharma and Professor Eki Sharma Oration with your presence today. Your participation has truly made the event meaningful. I would like to express our sincere gratitude to our honorable speaker, Professor Paramjit Khurana, for accepting our invitation and delivering the enlightening talk. Thank you, ma'am, for the wonderful lecture. I would like to thank Professor Shantanu Pal, head of the Department of Botany, for his wholehearted support and cooperation in organizing this oration in Archana Sharma Memorial Hall, Department of Botany, University of Calcutta. I would like to express our sincere appreciation to our distinguished guests and my colleagues from my department and other departments for their valuable presence. I also like to thank our guests who are attending this oration in online mode. I also want to thank our scholars and students for attending today's program. I would also like to thank all the university officials, teaching and non-teaching staff of the Department of Botany for all the infrastructural and organizational support. A special word of thanks is also due to Dr. Monika Bandyopadhyay and our scholars who have worked tirelessly behind the scene to ensure the smooth running of this program. Thank you for your continuous support and hard work. I'm incredibly grateful for your invaluable contribution. Last but not the least, I would like to thank Mr. Akash Mundol of Braindrops for the technical support to conduct the meeting seamlessly in online mode. Once again, thank you all for your unwavering support and participation. Thank you and have a wonderful day. Thank you.
Thank you. Thank you.
So I'd like to conclude our today's session with much thanks to our revered speaker and all the distinguished guests. Thank you.
Thank you so much.
And thanks to the department also and the Archana Memorial Foundation for organizing this excellent symposium. And we just heard something
Here we are
which is
also
very,
ending
very
our online
important.
meeting.
Thank you.
And
Thank you so much. I hope you can enjoy a cup of
Welcome.
tea together, all of you. Thank you so much.